Hello and welcome back to the Biochemistry for Health Sciences channel. Today we are going to talk about protein synthesis and transport. This is one of the most important topics for understanding medicine, especially for healthcare professionals of the future, as medicine is becoming more and more molecular. In previous videos, we have emphasized how important proteins are because proteins carry out most of the cellular activities of our 30 trillion cells in an adult human being. So, for example, nucleic acid binding proteins such as transcription factors, they regulate how fast our genes are expressed and therefore how fast our proteins are made in our cells. Enzymes make chemical reactions in our body go faster. Signal transduction proteins help cells respond appropriately to the changing environment. Transport proteins allow chemicals to get in and out of the cells as well as in and out of the organelles. Defense proteins such as complements and antibodies help protect us against viruses, bacteria, microorganisms, and of course help in wound healing. Structural proteins are required to maintain the structure of cells and tissues. And then there are many other proteins classes, what we call miscellaneous proteins, such as cytochromes that are involved in electron transport chain, proteins that inhibit enzymes, and proteins that carry chemicals such as iron, the protein ferritin. So since proteins do most of the cellular activities, therefore what a cell is actually doing can be determined if we know the protein profile, sort of a fingerprint of the protein profile. What proteins does a cell have and how much of each protein the cell has. So some proteins are made by all cells. So for example, the housekeeping proteins such as the glycolysis enzymes, these are required to generate energy. So these proteins are made by all kinds of specialized cells, the 200 different specialized cells that we have in an adult human being. Other proteins such as albumin is made only by specific cell types. So the liver hepatocyte makes albumin. The beta cell in the pancreas is the only type of cell that makes insulin. The chief cell of the stomach makes the enzyme pepsinogen, which is required for digestion. So basically the whole of anatomy and physiology is all about the 200 different cell types that we have in our body, and these functions of the cell types, what the cells do, eventually depends on what the proteins in the cells are doing. So the major steps involved in making a protein are shown here. So we have chromatin remodeling, then we have copying the DNA, the gene, into various RNAs, what we call transcription. And then these various RNA molecules undergo processing. All this happens in the nucleus. Then the various RNA molecules are transported out of the nucleus into the cytoplasm. And in the cytoplasm, we have protein synthesis or translation. So the proteins are made, and once the proteins are made, they undergo post-translational modifications, And eventually these proteins are then transported to the right location in the cell. So if a protein belongs to a membrane, the plasma membrane, the protein will be transported to the plasma membrane. If the protein works in the nucleus, then it will be transported to the nucleus, and so on. So eventually all of these proteins that you find in a cell gives the cell its specialization and its survival, and all of these cells eventually give us our anatomy and physiology. So in a way you can see the connection between anatomy, physiology, basically health, and if something goes wrong with health, then we are talking about diseases, pathology. So eventually normal anatomy and physiology and abnormal anatomy and physiology are all related to protein profiles of cells. So let us look at an example, look at the various steps of how, let's say, a simple protein, let's say this protein here, methionine, phenylalanine, proline, valine, lysine. So you remember this is amino acid number 1, number 2, number 3, number 4, number 5. So let's look at how this protein is made, let's say by a liver cell. The information to make this protein is in the gene M found in the DNA of your nucleus. And let's say this protein is made in response to high levels of a hormone, which is a signal that appears in our bloodstream after a meal. So let's look at how your liver cell is going to make this protein. Look at the various steps. As healthcare professionals and students, we don't have to know all the details. We have to look at the bigger picture, and that's what we'll do in this video. Okay, so we have a meal, and because of the meal, some hormone is released. And of course, the hormone travels in your bloodstream, and when it reaches the liver, the hormone basically gets out of the blood vessel and travels towards the liver side. It diffuses. Okay, so diffuses towards the hepatocyte. Once it's near the hepatocytes, the hormone is then going to bind the receptor. So these blue structures here are the receptors, and you can see the hormone binding to the receptor. And we'll talk about this more of cell signaling in future videos. But once the hormone or the signal binds the receptor, that triggers a signal transduction pathway. And this pathway can lead to the activation of various cell-specific transcription factors. So in this case, let's say the transcription factors have been activated and they enter the nucleus. Here the transcription factors will now bind to the nucleus, and this will start the process of protein synthesis by triggering transcription or RNA synthesis. So before we talk about the first step of protein synthesis, which is chromatin remodeling and transcription, let's look at this gene, the gene of a protein. Let's look at some of the basic structural features of these genes, the protein coding genes that we find in our nucleus, in our DNA. So here are our two DNA strands. Here are the two DNA strands. So for making a protein, we'll just copy one of these strands. Let's say this strand is running 3' in this direction and 5' in that direction. So we're going to copy this strand. We call this strand that we are going to copy, we call this strand the template strand. The template strand. So let's say the template strand is running 3' to 5' in this direction. The template strand is also sometimes called the antisense strand, non-coding strand, the minor strand, and so on. Okay, so our template strand is running 3' to 5'. We are going to copy this strand into RNA. Okay, and that is going to be done from 5' to 3'. So the first nucleotide we copy or transcribe will be the +1, and then +2, +3, and so on. This direction going down the gene is called downstream, and then the opposite direction here where we label the nucleotides as -1, -2, -3, -4, -5, and so on. This is the upstream direction. So this is here the message that we need to make that protein, our simple protein. This is really the message we need. But notice that this message has got something useful, the exons. And then between the exons, this message is separated by some sequences that are not really necessary to make the protein. Okay, these are called introns. So it's very common, most of our genes have useful message that is required to make the protein. And then these useful message segments called exons are separated by one or more introns. A few more important words about this gene M. This gene M has also sequences that we call promoters and enhancers. So for example, here there is a Tata box. This is a very common and important promoter for many genes, located somewhere around in the -25 direction. If this is your +1 where we start the transcription. And then there is more promoters, regulatory promoters such as your GC, CAT sequences found around -100 and around -80. And then we have special sequences called enhancer sequences. And these enhancer sequences can be upstream or downstream and can be very far away from the gene that carries the information to make the protein. Okay, now that we know what gene M looks like, let's see what happens after the hormone binds the receptor, and that sends the transcription factors into the nucleus, and now the transcription factors are going to bind the gene. So let's see what happens next. Okay, so the transcription factors have entered the nucleus and they are going to bind the various parts of the gene. For example, they're going to bind the regulatory promoter sequences, and they're going to bind the enhancer sequences. Notice also that the genetic information to make the protein, it might be blocked by proteins called histones. Okay, so remember our DNA has a lot of histones that are bound to the DNA. So these structures here shown are histones. So after the transcription factors bind the enhancers as well as the regulatory promoter sequences, what happens is the DNA is bent. And this DNA bending is done by what we call as DNA bending proteins. And one example of DNA bending proteins are the high mobility group proteins or HMG proteins. There are many other types of DNA bending proteins, but we will just focus on a few examples without getting into too much details. By that bending of the DNA, now we see the enhancer sequences which can be very far away from the gene. So now we see the enhancer sequences get very close to the promoter sequences of the gene. So in the next step, there are a variety of helper proteins. So there are many different types of helper proteins that are going to prepare the promoter region, going to get it ready for transcription, get it ready for the RNA polymerase to come and bind to the promoter region and start copying the gene. So these proteins include chromatin remodeler proteins. Basically, the chromatin remodeler proteins, they remove the histones from the DNA so that this genetic information is accessible, can be read. And this removal can be done by dislodging the histones, even changing the types of histones that are attached to the DNA, and more commonly by removing the positive charges on the histones. So histones have a lot of amine groups which when it's protonated, remember they are positively charged. So by chemically modifying these amine groups by acetylation and other kinds of chemical reactions, this positive charge is removed. And when the positive charge is removed, remember DNA is negatively charged. So without the positive charges, this attraction goes away and the histone proteins get easily removed from the DNA, thereby getting the genetic information exposed for transcription. Besides chromatin remodeling, the other proteins such as mediators or co-activators and a variety of basal transcription factors such as transcription factor 2D, 2A, 2B, 2F, and so on. These proteins will all come and bind close to this region of the promoter, and basically they will set up a nice platform so that the RNA polymerase can easily search for this platform and bind appropriately to this platform to be near this region so that it can start copying the DNA template. And that's exactly what happens once the platform is made by these mediator proteins and transcription factors. Now the RNA polymerase can basically bind to this region and start the process of copying the DNA. So RNA polymerase 2 is the one we'll focus on in this video. This will copy the DNA template into mRNA. RNA polymerase 1 copies ribosomal RNA, while RNA polymerase 3 copies mostly transfer RNA as well as a few other different types of RNA. All these different RNA polymerases, they have their own promoter sequences and enhancer sequences. So now the RNA polymerase is in the right location on the DNA next to the information to be copied. Various transcription factors help the RNA polymerase to carry out this function. So for example, TF2H has enzyme activity that can unwind the two DNA to form the transcription bubble. So this strand and this strand is unwound, and so that the RNA polymerase can travel through the DNA. TFA2H kinase, another transcription factor, will phosphorylate the RNA, add phosphate groups to RNA polymerase 2 to help it change shape so that it can start copying the DNA by transcription. And before it starts copying the DNA, there are other proteins such as SPT5. These proteins make sure that the DNA template strand is not damaged. So if it's okay, then it allows the RNA polymerase to start moving to copy it. If it's not okay, if the DNA is damaged, then the RNA polymerase is not allowed to proceed, and it may even be destroyed by the cellular machinery. Okay, so once the checkpoint is passed, then we are ready to start copying the DNA in a phase that we call transcription elongation. So now the RNA polymerase is ready to copy the DNA template strand. It starts moving. Most of the transcription factors that were bound to the RNA polymerase are also removed from this RNA polymerase, and it starts now copying the DNA template strand. So a T on the template DNA will be copied as an A on the mRNA. A on the DNA is copied as a U, a C is copied as G, and A as U, AU, and so on. So as it copies the template, it may stop at many more checkpoints, and there are all kinds of proteins to make sure that the copying is accurate and the DNA is not damaged. So the template strand keeps getting copied by the RNA polymerase, and there are many other proteins called transcription elongation factors. Examples of these are shown here. And these transcription elongation factors help the RNA polymerase copy the template strand during the elongation phase of transcription. So once we have about 20 or so nucleotides copied, we have a small mRNA transcript. Then what we have here the 5' end. There is the enzyme called 7-methylguanosine cap synthesis enzyme. This will now bind the RNA polymerase, and it will attach a 7-methylguanosine cap at the 5' end of this newly formed messenger RNA. This is called capping, and is one example of what we call RNA processing that happens as soon as the mRNA starts getting made. Then once the cap is formed, there is a cap binding protein that brings the cap, a cap binding protein that brings the cap close to the RNA polymerase during elongation. So as elongation happens and we copy not only the exons but also the introns shown in green, so another processing happens, and that's called splicing. Basically the intron, one or more introns that were copied in the mRNA, they are removed either by self-splicing or by the use of these huge protein complexes called spliceosomes. So the introns that are copied in the new mRNA, they get removed as elongation continues. And once the introns are removed, the two exons are correctly joined together, once again, either by self-splicing systems or by the use of spliceosomes. So in this way, the whole template is copied, and at the end of the template, there are special sequences, termination sequences, and these sequences are also copied on the mRNA, and these termination sequences are recognized by special proteins called cleavage and polyadenylation specificity factors, or CPSF, as well as other proteins. So this CPSF will bind to the 3' end and it will break off the 3' end of the mRNA right here. So this will break off and the 3' end of the mRNA will now be free. So here we see the 3' end of the mRNA. Remember, the 5' end is connected to the cap binding complex, still attached to the RNA polymerase. Now that the 3' end is free, we now have the next step that happens, and that's the addition of a 3' tail to the mRNA. And this is done by the enzyme polyadenylate polymerase. So this adds a poly A tail, so basically A, so on, a poly A tail to the 3' end, which is another example of RNA processing. So capping is an RNA processing, adding a tail is an RNA processing, splicing is RNA processing. So basically RNA processing is happening, most of this RNA processing is happening during transcription. So now we have the matured mRNA transcript. We have the cap. This is the untranslated region, UTR, 5' UTR. This is the 3' UTR on the other end. And we have the message necessary to carry out the synthesis of the protein. So now we have our mRNA. So in a similar fashion, we make all the ribosomal RNA, we make all the transfer RNA necessary for protein synthesis, and all of these RNAs are transported out of the nucleus through nuclear pores. So they are actual pores in the nucleus. So they are transported out by special transport proteins, and all of this RNA now is are in the cytoplasm. Okay, so transcription is all done. All the necessary RNAs have been processed and transported to the cytoplasm. Now we are ready for protein synthesis. So before we start, the liver cell starts protein synthesis, it has to attach the right amino acid to the correct transfer RNA. So for example, methionine has to be attached to the transfer RNA that carries an anticodon UAC. Okay, so the right amino acid has to be attached to the right transfer RNA. So we have about 50 or so transfer RNAs. And remember, there are about 20 amino acids that make up proteins. So doing this attachment is not easy. It's quite a complex task and has to be done accurately. And this is done by the enzyme called aminoacyl tRNA synthesis. So basically you can see the carboxyl group here reacts with ATP of the amino acid carboxyl reacts with ATP. The carboxyl gets activated or reactive because there's an AMP attached to it. So we have an amino acid AMP made. Pyrophosphate is released. This drives the reaction to the right. And then in the next step, the sugar OH of the transfer RNA here will attack this carboxyl. The AMP leaves. So this carboxyl is reactive because it has an AMP attached as a leaving group. And now we have the amino acid attached to the sugar of the transfer RNA through an ester linkage. So the amino acid is attached to the transfer RNA via an ester linkage. These transfer RNAs that have amino acid attached, we call this transfer RNA charged transfer RNA. This charge has got nothing to do with positive, negative. Charge here simply means there is an amino acid attached to the transfer RNA. Okay, the transfer RNA that doesn't have an amino acid attached, that's called uncharged transfer RNA. So this is how the correct amino acid will be attached to the correct transfer RNA by this remarkable enzyme called amino acyl tRNA synthesis that of course requires energy. So in the next step, we form what's called the pre-initiation complex. So this requires the help of proteins called initiation factors. So translation initiation factors. So you can see the tRNA carrying methionine gets attached to the small ribosomal subunit, the 40S subunit. And initiation factors prevent binding of other tRNAs to this subunit, prevents the bigger subunit from joining. They help create the mRNA entry channel, and it helps bind the methionine tRNA, helps this bind to the 40S at a correct location, what we call as the P site. So all of these activities are done with the help of initiation factors to form the pre-initiation complex between the transfer RNA and the 40S, the small ribosomal subunit. So once the pre-initiation complex is formed, we have the tRNA carrying methionine attached correctly to the 40S, small ribosomal subunit. Now we are ready to attach our mRNA to this complex. So here our mRNA now gets attached, and the pre-initiation complex now becomes the initiation complex. So notice we again need the help of initiation factors. Not only do they help attach the mRNA to the 40S small unit, but importantly also they make sure that the AUG, the start site, the AUG is located and correctly placed on the ribosome. So this AUG site is recognized by intrinsic factors, what we call ribosomal scanning. By looking at a certain sequence, we call it the Kozak sequence. This Kozak sequence is near the 5' UTR. So somewhere near here, 5' UTR is the Kozak sequence. And intrinsic factors help recognize this sequence and make sure that the mRNA is bound correctly to the 40S subunit. So the P site now has the methionine tRNA. The A site is being blocked from any tRNA binding by the intrinsic factor. So more intrinsic factors help in preventing the A site from binding any transfer RNAs. Okay, so once the initiation complex is formed, we are now ready to attach the larger ribosomal subunit. So with the help of intrinsic factors, other intrinsic factors, the largest ribosomal subunit, 60S, will bind to the smaller 40S. So the 60S will bind to the smaller ribosomal subunit to form the complete ribosome, which we call the 80S ribosome. Notice that a lot of GTP is used by these proteins. Okay, now that we have the two subunits and the mRNA bound, we are now ready to get the next transfer RNA. So if you look at this codon, AUG, according to genetic code, codes for methionine. UUU, according to the genetic code, codes for phenylalanine. So UUU is the codon, so the anticodon that binds UUU would be AAA. So we need an AAA anticodon transfer RNA that is charged with phenylalanine. We need that to come and bind to this site. And that's what happens with the help of elongation factors. The phenylalanine transfer RNA is brought to the A site next to the P site carrying the methionine transfer RNA. And so you see the codon interacting with the anticodon. Anticodon interaction, which is regulated or controlled by the 40S subunit. As GTP is hydrolyzed, this transfer RNA gets very close to the methionine carrying transfer RNA. Okay, so now we have the two transfer RNA next to one another. Now we are ready to react. We are really ready to react these two amino acids to form the peptide bond. So now we form the peptide bond. Basically, the free amine of the phenylalanine will react with the ester that joins methionine with its transfer RNA. And this reaction leads to the formation of a peptide bond between methionine and phenylalanine. This reaction, this actual peptide bond formation reaction, is catalyzed by the 60S subunit ribosomal RNA. So the catalyst here, the enzyme here, is not a protein, but it's an RNA. But there are proteins, elongation factors that help this 60S RNA catalyze this reaction between the two amino acids. So after this reaction, what we have on the P side is an uncharged transfer RNA. What we have on the A side is the transfer RNA that carries the dipeptide, the two amino acids joined together, and here is an E site which is still has nothing in it. Okay, so now we are ready for the ribosome to move in this direction, and when that happens, this empty transfer RNA will come to the E site, while the dipeptide transfer RNA will come to the P site, and that will leave the A site empty, and that's exactly what happens. So now the A site is empty, the dipeptide is on the P side, and the uncharged transfer RNA is on the E site. So notice once again we require energy for all of these processes, and most of the energy is used is in the form of GTP. So GTP, besides ATP, is another important chemical form of energy that cells use. So with the help of the energy, now we are ready to get rid of this empty, this uncharged transfer RNA. So by changing the shape in the help of release elongation factors, this uncharged transfer RNA is removed from the E site. So now we have the dipeptide at the P site, while the E site is empty and the A site is also empty. So now that we have the A site empty, we are ready to accept the next transfer RNA. And since the next codon is CCC, we need a GGG anticodon transfer RNA. And according to the genetic code, CCC represents the amino acid proline. So we need a proline transfer RNA to come to the A site in the next step. And that's exactly what happens. The proline transfer RNA will come and bind to the A site. Once again, we need elongation factors to help do this. So now, once again, we will have a transfer RNA attached to an amino acid on the A site. So these steps now will be repeated, and elongation will keep on happening until all of these codons are read. So as the ribosome move and reach all the codons, now we get to the end of the message of the mRNA, and here there is what we call a stop codon. There are 3 stop codons, UAA, UAG, UGA, according to a genetic code. And these stop codons are recognized by proteins called release factors. So these release factors will recognize these stop codons. And then these release factors will help break the connection, the ester connection between the polypeptide chain and the transfer RNA. So this ester cleavage will release the polypeptide chain. So the newly formed polypeptide chain will get out of the ribosome through the protein exit channel. So there is this channel, and this protein will now get out of the ribosome. The release factors also help dissociate the 60S from the 40S, and the mRNA is also freed from the ribosome. So this is how we get our protein. So here we have the protein with an amine terminal, methionine, phenylalanine, next amino acid number 2, number 3 proline, number 4 valine, and the C terminal amino acid lysine. So remember, this information came from the mRNA, because mRNA is nucleotide, the language here is nucleotide, this is amino acid, a different language. So that's why we call this process translation. Translation. We actually take one language, nucleotide language, and make it into amino acid language of protein. So that's what we call protein synthesis. Protein synthesis is called translation. So once the protein is made, or while it is being made, it can be modified. If it's modified while it is being made on the ribosome, we call it co-translational modification. Or if it's modified after it is made, released from the ribosome, we call it post-translational modification. So proteins can go through many modifications. Proteins are folded into their correct 3-dimensional shapes. That's done by the help of other proteins called chaperones. All newly made proteins have methionine at the end terminal, but that methionine may be removed by proteases in certain proteins. Newly made proteins have signal sequences at the end terminal. These signal sequences tell the cell where the protein should go. Should the protein go to the cell membrane? Should the protein go into the mitochondria? And so on. So these signal sequences help tell the cell what will be the location of the protein. And many chemical reactions, covalent modifications can happen to various different types of proteins. There could be phosphorylation, phosphates could be added to proteins, to certain amino acids. Hydroxyl groups could be added to certain amino acids, hydroxylation. Methyl groups could be added, methylation. Acetyl groups could be added, carboxylation, carboxyl groups added. Disulfide bonds form between cysteines. Glycosylation, some proteins have carbohydrates attached to them. Other proteins may be connected to lipids. Metal ions may be attached to proteins. Organic chemicals such as hemes may be attached to proteins. So various proteins have various kinds of modifications that happen. And as we go through different videos, we will see different proteins with various modifications. So shown here in this diagram is a beta cell of the pancreas that makes the hormone insulin. So as you can see, insulin is made in the cytoplasm. In this case, the signal sequence shown in the red here tells the cell that insulin should be secreted out of the cell. So what happens is this insulin enters the ER. There the signal sequence is removed. Disulfides are formed, and then it gets transferred to the Golgi. And in the Golgi, one segment of the insulin is removed. This is called the C peptide or the C chain. So the C chain is removed, and the insulin basically is your A and B chain. So the C chain has been removed. So this AB chain forms the insulin. So this AB chain insulin formed together with the C peptide is then transported as vesicles. So these vesicles is what stores your insulin in the beta cell. Once your blood glucose is high, and that happens after a meal, that glucose will then trigger a cell signaling which will lead to the increase of calcium ions in the beta cell. And these calcium ions will help trigger the binding of these vesicles to the plasma membrane. And that causes the exocytosis, release of the insulin, as well as the C peptide, which then both of which get out into the bloodstream. So that's how your insulin is synthesized and secreted after a meal. And we'll talk more about this in future videos on cell signaling. So just to generalize, protein transport, protein synthesis happens on ribosome. Protein is folded. Protein is sent to various parts of the cell by the help of signal sequences. Those proteins that are meant for secretion to work in the ER or the Golgi or lysosome, these are recognized by special transport proteins. They are brought to the ER, endoplasmic reticulum. In the ER, carbohydrate, some proteins have carbohydrates attached. These are called glycoproteins. So one of the places where carbohydrates is attached is in the endoplasmic reticulum. So carbohydrates can be attached. The protein synthesis, for those that go to the ER or the membrane or the Golgi, they complete their synthesis in the ER. Those ER proteins that are for ER stays in the ER. The others are transported out of the ER. So you can see the transport out of ER. Then these vesicles here will take these proteins to the Golgi, the next step. And in the Golgi, more carbohydrate may be added to glycoproteins. So those proteins that are meant for the Golgi, they will stay in the Golgi. But those proteins that are going to go to lysosomes, they will be transported to the lysosomes. And those that go to the plasma membrane, they will go to the plasma membrane. They become these membrane proteins. And those that are to be secreted like insulin, those membrane vesicle proteins will fused with the plasma membrane, and the protein comes out by exocytosis, as we saw in the case of insulin. So there we have it. That's how we make proteins in the cell that are required by the cell, and then these proteins are transported to the right location in the cell. For example, the Crab enzyme proteins will be in the mitochondria, the glycolysis proteins will be in the cytoplasm, proteins required for activities in the nucleus, such as your RNA polymerase, will go to the nucleus, and so on. So this is a long story, but it's a very, very important story, how protein is made and transported for healthcare professionals, because proteins carry out most things for cells. So if something goes wrong in one or more of these steps of synthesis and transport, that can lead to diseases. So many diseases today can be explained by defects in one or more steps of protein synthesis and transport. Let's look at one example, cystic fibrosis. Cystic fibrosis is a disease where we have problems with chloride transport because the protein that is involved in cystic fibrosis is a protein called CFTR, cystic fibrosis transmembrane conductance regulator, and this protein is involved in the transport of chloride ions across cells, which is indirectly related to transport of sodium and therefore transport of water. So patients with cystic fibrosis may have one or more defects in these steps of protein synthesis and transport. So for example, in type 1 mutations, some people may have problems with the actual transcription step, the synthesis of mRNA. In type 2, which is the most common that we see in many patients with cystic fibrosis, the problem is right here, the transport of the protein. So the protein is not transported correctly to the membrane. That trafficking doesn't happen correctly. And so the patient doesn't have CFTR in their membrane. In type 3 mutation, there is CFTR on the membrane, so it is there, but the chloride channel gate doesn't operate correctly. It doesn't open correctly when stimulated. So although the protein is there, it's not functioning correctly, and that leads to impaired chloride transport, sodium transport, eventually water transport. In mutation type 4, there is the channel gate does open, but the channel conductance is not very good. The chloride flow is not fast enough that it leads to problems with sodium eventually and water transport. In mutation 5, which some people have, there is the protein in the membrane, but there is a reduced number of CFTR proteins. Okay, so if an average person had 20,000 CFTR proteins on a membrane, this patient may have only 5,000. Okay, and in mutation 6, class 6, the protein is there but is unstable and therefore doesn't work very well. So you can see from this example that a disease such as cystic fibrosis may be due to one or more defects at different stages of protein synthesis and transport. And also why this is important, because now pharmaceutical companies are developing drugs that can correct one step. So as a healthcare professional, you need to know these steps so that you can explain how the drug is working and whether that drug is necessary for that patient because that drug is very specific for that step. Because in your patient, you may not have that step that is defective. Maybe another step is defective and the drug may not be effective for that step. Okay, so as we are getting towards personalized medicine and molecular medicine, it's becoming more and more important to understand diseases at the molecular level, and this is why protein synthesis and transport is becoming one of the most important topics in medicine today. So cystic fibrosis is just one example. There are, like I said, there are many diseases out there. Cancer is another big one. Cancer, understanding cancer, various types of cancer at the molecular steps is becoming very, very important because once again, the new drugs that are used for cancer treatment are very specific with their steps. Okay, very specific. So you need to understand the biochemical steps involved in cancer, in diabetes, in cystic fibrosis, and all kinds of diseases in order to understand how to treat the disease. Okay, so before we end this video, after reviewing this video, I saw that under the translation, instead of saying initiation factors a few times, I used the word intrinsic factor. Okay, intrinsic factor. So just to clarify, it should be initiation factors, not intrinsic factor. Some of you might know that intrinsic factor is a protein that helps us absorb vitamin B12. So that has got nothing to do with this video. All right, so that's all for now. Goodbye and have a wonderful day. Hopefully you would subscribe to this channel where you will learn a lot about the connections between the world of chemistry as well as the world of medicine. Bye-bye.